Hello everyone, welcome to my YouTube channel. I hope you all are fine. So today's topic is diffusion potential, resting membrane potential and action potential. Let's talk about the ion channels first. So remember that in the first video we talked about the structure of cell membrane and the two types of proteins, the integral proteins and the peripheral proteins. So ion channels are nothing but these are the integral proteins that span the membrane, and when they open they permit the passage of certain ions. So these ion channels have certain characteristics. So ion channels are selective. This means that they permit the passage of some ions but not others, and this selectivity is based upon the size of the channel and the distribution of charges that line it. So let's say we have a small channel and it is negatively charged, so it will be selective for small cations and exclude the large solutes and anions because it is itself negatively charged, so it will not allow the anions to pass through it. And also it is small in size, so it will allow only the small solutes to pass through it. And on the other hand, if we have a small channel that is lined with positively charged groups, then it will be selective for the small anions and will exclude the large solutes and cations. And next property we have is that the ion channels may be opened or closed. So if the ion channel is open, then it will allow the ions to flow through it selectively because it is selective. So it will allow only selective ions to flow through the channel. And when the channel is closed, the ions cannot flow through this. Simple. And the third thing we have is the conductance of a channel. That simply means the permeability of a channel. So if the channel is open, the higher is the conductance and vice versa. Okay, so the opening and closing of channels is controlled by gates. I have discussed it in my previous video that either the channels are voltage gated or they are ligand gated. Voltage gated means that the channel will open whenever there is a difference in the membrane potential. And the ligand gated channels, they are opened or closed by certain chemicals like hormones or second messengers, neurotransmitters. When they bind with the receptors, this causes the opening of certain channels, and this will allow the chemicals to flow through this to the inside of the membrane. And whatever is the mechanism, we will discuss it. Okay, so here just for a basic concept that how the voltage gated channels work. The sodium channel, it has two gates, activation and activation gate. When both the gates are open, only then the channel is permeable to sodium. But if either of them is closed, then we consider it impermeable to sodium. Okay, the next is the ligand gated channels. These are opened or closed by hormones, second messengers, or neurotransmitters. For example, we have the nicotinic receptors for acetylcholine. So what happens that this nicotinic receptor for acetylcholine at the motor end plate, it is an ion channel that opens when acetylcholine binds to it. So it is an example of a ligand gated channel because when acetylcholine binds, only then it opens. And when open, it is permeable to sodium and potassium, and this causes the motor end plate to depolarize. So just for a basic concept of a ligand gated channel, we will talk about it in detail in the upcoming videos. Okay, so before talking about the resting membrane potential and action potential, let's talk about the basic physics of membrane potentials. So we have diffusion potential and we have an equilibrium potential. So diffusion potential, it is caused by an ion concentration difference on the two sides of the membrane. Okay, in order to understand this, let's take the example of a nerve fiber membrane. So we know that the potassium concentration is great inside the membrane, whereas it is low outside the membrane. So we assume that the membrane in this instance is only permeable to potassium ions, but it is not permeable to any other ion. And because of a large potassium concentration gradient from the inside towards the outside, there is a strong tendency for the extra number of potassium ions to diffuse outward. So as the potassium ions, they diffuse outward, they carry the positive electrical charges to the outside of the membrane, thus creating electropositivity. outside and electronegativity inside the membrane, because the negative anions that remain behind, they do not diffuse outward with potassium, because we assume that the membrane is only permeable to potassium ions. So inside the membrane there is electronegativity, whereas outside, because of diffusion of potassium ions, the electropositivity develops. And a point comes when the diffusion potential of potassium, it becomes strong enough and it becomes equal to the concentration gradient. Now the electrical gradient, it becomes equal to the concentration gradient, and there will be no net movement of ions across the membrane, despite the fact that there is still this potassium gradient present, and also the membrane is fully permeable to potassium, but there is no net movement of potassium to the outside of the membrane. So equilibrium comes in, and now we call it as equilibrium potential. We do not call it diffusion potential of potassium, but now it becomes the equilibrium potential or the Nernst potential, because the equilibrium potential is also known as the Nernst potential. Okay, let's reverse the situation, and now we consider that the membrane is highly permeable to sodium ions only and impermeable to all other ions. And sodium, it is higher outside the membrane. The concentration of sodium is higher extracellularly. So diffusion of positively charged sodium ions to the inside, it creates a membrane potential of opposite polarity to that of potassium. That is negativity outside the membrane and positivity inside. Again, as we talked about in the potassium diffusion potential, that the membrane potential rises high enough that there will be no further net diffusion of the sodium ions to the inside of the membrane. The equilibrium comes in, and this stops the further diffusion of sodium ions. So for potassium, the diffusion potential is 94 millivolt with negativity inside the membrane. But in case of sodium, we have the diffusion potential that is 61 millivolts, but with positivity inside the membrane. So that is the difference between these two. Okay, let's see what's written in the BRS Physiology. So diffusion potential is the potential difference that is generated across a membrane because of a concentration difference of an ion. So the size of the potential, it depends upon the size of the concentration gradient, and the sign of the diffusion potential, it depends upon whether the diffusing ions is positively or negatively charged. So the sign, like in case of the potassium diffusion potential, it is negative, whereas in the sodium diffusion potential we discussed that it is positive. Minus 94 millivolt for potassium and plus 61 millivolt for sodium. Then we have the equilibrium potential or Nernst potential. It is the potential difference that would exactly balance, oppose the tendency for diffusion down the concentration difference. The potential difference when there is no further net diffusion of the ion. This is basically an electrochemical equilibrium. So at electrochemical equilibrium, the chemical and the electrical driving forces that act on that ion are equal and opposite, and there is no further net diffusion of the ion occurs. So here in the BRS Physiology, they have an example of two solutions of sodium chloride, and they are separated by a membrane. So in the example 1, they are saying that it is permeable to sodium only, but not to chloride. And also the concentration of sodium chloride in the solution 1, it is higher than that of solution 2. So what happens that sodium will diffuse from solution 1 to solution 2, from higher concentration to low concentration, down the concentration gradient, and chloride is impermeable, and therefore it will not accompany the sodium. And as a result of this, a diffusion potential will develop across the membrane, and solution 1 will become negative with respect to solution 2, and as time passes, the potential difference becomes large enough to oppose further net diffusion of sodium. The potential difference that exactly counterbalances the diffusion of sodium down the concentration gradient, we call it as sodium equilibrium potential. And for the chloride diffusion potential, everything is same, but now the membrane is permeable to only chloride rather than to sodium. and the only difference would be that now the diffusion potential will be positive, because chloride is diffusing from solution 1 to solution 2, and sodium remains behind. So the diffusion potential becomes positive, whereas in the previous example the diffusion potential was negative. And again, when the diffusion potential of chloride becomes large enough, the electrochemical equilibrium develops, and the electrical and chemical driving forces on the chloride are equal and opposite, that there is no net diffusion of chloride. And then we have this Nernst equation to calculate the equilibrium potential. That is the potential at which the ion would be at the electrochemical equilibrium. So here it is calculated for sodium, that is plus 60 millivolt. And on the next page we have the approximate values for equilibrium potential in the nerve and skeletal muscles. So remember these for sodium, calcium, potassium and chloride. So we talked about the basic physics of membrane potentials, and that will help you understand the concept of resting membrane potential and action potential that I will talk about in the next video. So like, share and subscribe to my channel. Thank you so much for watching. See you in the next video.